So much. So hi everyone, my name is Feng and I'm from the University of Michigan. And actually I was having the question if I was like assigned to the wrong track because my talk will focus more on a specific topic instead of like a career or facility. But I would still appreciate the opportunity and I hope our resource can help more like BioInfo-Core research community. Okay, so let's start. As we know, gene transcription is regulated by the cooperation of enhancers and promoters. And, oh, okay. It's fine. Yeah. Yeah, good. Okay, thank you so much. So it is not only related to the properties of enhancer promoters, also controlled by their direct communication in the 3D space. Therefore, people are paying more and more attention to like 3D chromatin organizations recently. Therefore, requires technologies for accurately capturing those 3D chromatin conformation. And Hi-C is a common tool for measuring the 3D proximity between pairs of genomic regions by like cross-linking, cutting, and high-throughput sequencing. And actually, the result can be visualized as contact maps. And a pixel on that contact map it indicates the interactions between the two anchor regions, like labeled in that figure. However, although Hi-C has helped us uncover like the hierarchical structures of chromatin organization, it is still rarely utilized in analyzing transcription regulation. The reasons are quite intuitive. First, the typical lengths of regulatory elements like enhancers, promoters, and binding motifs. Okay. Okay. Yeah, that's right. Does it work now? Okay. So where are we? Okay, the major reasons why Hi-C is still rarely utilized in analyzing transcriptional regulation. First, the typical lengths of like those regulatory elements like enhancers, promoters, they're usually tens to hundreds of base pair. However, Hi-C can only reach a resolution of kilobase level. And second, most of enhancer promoter interactions are just short range and within topological associated domains. So they are not well captured by Hi-C. But recently, a new technology, micro-C, by cutting the DNA into mononucleosomes, it can reach a much higher resolution and outperforms Hi-C at detecting short-range interactions. Actually, micro-C contact maps can be analyzed at 200 base pair resolution, and a lot of interaction between enhancer promoters and more fine scale structures can be detected by micro-C. However, current micro-C contact maps are only available in 3 cell lines, H1 embryonic stem cell, HFF, and WTC11. And this is where our motivations come from. Can we use deep learning models to impute more nucleosome resolution contact maps for like different tissues and cell lines? And of course, the input should be at least nucleosome resolution and should be related to 3D chromatin organization and transcription regulation, and also widely available. Therefore, we chose chromatin accessibility, TF binding, and histone modifications. And this is our model Caesar. The input, the key input is 6 cell type specific epigenomic features, ATAC6, CTCF, and a couple of histone modifications. And it also requires a low resolution Hi-C contact maps, but it is not that important in like predicting short range interactions. So we can use a human average Hi-C. And the output is the cell type specific contact map at 200 base pair resolution, in which you can observe like more detailed structures in a region of your interest. And here are some example inputs and outputs. We can see like Hi-C, Caesar prediction result, and micro-C. Caesar can predict some fine scale structures that are undetected by Hi-C, like polyform regions, short range loops and stripes. And also in our validation experiment, we split human chromosomes into 3 sets, train on the train set, tune the parameters on the tune set, and evaluate our performance on the test set. we calculated the correlation coefficient between our prediction results and the real high-resolution micro-C content maps. And of course, we can observe our prediction result matches better with micro-C than the original low-resolution high-C. And also, we compared our model with two baseline methods, which also predict content maps, which is high-C plus and high-C reg, and Caesar outperforms those two baselines. In a further experimental result, let's just focus on the cross-cell type experiment, which we train the model on one cell line and test on another cell line. We found that this model can still achieve the good performance. Therefore, it indicates that we can use our model to predict the nucleosome resolution content maps for various, more diverse tissue and cell lines. And actually, Caesar can also predict more than 60% of fine-scale intra-TET loops and stripes from micro-C content maps. And as we mentioned before, transcriptional regulation is highly related to 3D interactions between genomic regions. And we could also validate our model by checking whether it can predict this kind of interactions. And we use CRISPR-I to locate the gene enhancer pairs. I'll skip the details here, but we only need to know the peaks of CRISPR-I scores usually correspond to enhancers and promoters. And in the example regions, in K562 and kappas, Caesar can predict the interaction between MYC gene and its enhancer in PVT1, which Hi-C cannot. And similarly, in K562, Caesar predicts the interaction between GATA1 and its key enhancer in HDAC6. So this demonstrates that our model can be used to predict the interaction between genes and its key regulatory elements in various cell tissue and cell lines. So in the end, we imputed 91 nucleosome resolution content maps from only public ENCODE data. And those content maps are all available, published online on our website, in which you can browse on the website to locate the regions you are interested in and check the enhancer promoter interactions which you are interested in. And for the more details of our models and our experiments, you can refer to our paper on Nature Communications. And that's the end of my talk. Thank you so much for your attention. Yeah.
Thanks for this really interesting. I'm aware that it is better to predict the interactions in 3D, but did you also compare your results to, I don't know, high-scale CRISPR experiments or ABC models or
it's a little different than pure ABC models because we just focus on 3D genomics. And I think it might be because if I remember that correctly, ABC model can only be calculated in a couple of cell lines because only those cell lines have the highly sequenced high-C content maps. I think I hope our model can benefit those people who are interested in 3D genomics but focus on a tissue or cell line which we don't have many resources. Yeah, so basically it's a compensate or not a compensate, just a resource. Yeah. Does that answer your question?
Yeah.
Thank you so much.